the Western College of Veterinary Medicine in Saskatoon. And I'm also a veterinarian, and this is my lab. Our supervisor is Dr. Elamir Simko. And most of the members of our lab are either veterinarians or veterinary students, and many of us have training in veterinary pathology. We're a very international group. We have students from Brazil and Ukraine, as well as Canada. And we're also a very fun group and a good team, because certainly with honeybee research, your research is only as good as your team. We're also very lucky to have great support from the Saskatchewan beekeepers. So this is the Saskatchewan Beekeepers Development Commission booth at IPM, and we're very proud to stand shoulder to shoulder with them because they certainly have supported our research from the start and continue to do so. So today I'm going to be speaking about European Foulbrood disease, which seems to be a disease that is becoming more common. Just talking to provincial apiculturists in Western Canada, it seems like this disease is on the rise. As well, there seems to be an association between European Foulbrood and colonies that are pollinating blueberries in BC. And so because we know this is a disease of stress, we wanted to investigate the predisposing factors for European Foulbrood a little more closely. So what is EFB? So it's a bacterial disease of honeybee larvae caused by the bacterium Melissococcus plutonius. It's typically a disease of early spring, which will commonly appear after the first nectar flow. So the nectar in the environment will stimulate the colony to rear brood, but oftentimes in early spring, the colony does not have an adequate number of nursing bees to care for that brood, and that can predispose the colony to disease. Luckily though, as the nectar flow continues, many colonies are able to spontaneously recover from this disease on their own as they acquire a stronger nursing bee population. So how does the disease work? Well, the pathogenesis is incompletely understood, but this is more or less our best guess. So a newly hatched larva will be infected by the brood food provided by a nurse bee. The bacteria will then proliferate in the midgut of this infected larva. And then under conditions of colony stress, for example, reduced nectar in the environment, the bacteria will outcompete the larva for food, resulting in larval death from starvation. Secondary bacteria can then come in and invade this dead larva, and the dead larva will hopefully be removed from the colony, thus breaking the disease cycle, and the colony will recover. Alternatively, when the colony is not stressed and there's abundant nectar in the environment, sometimes these infected larvae can survive, but they may emerge as smaller adult worker bees. And these subclinically infected workers can then spread the bacteria throughout the colony, but there will be no clinical evidence of disease in the colony. So what are the clinical signs of EFB? So this is a brood frame that was mailed to me last summer and cultured positive. So we can see that there is a shotgun brood pattern here. And if we look closer, there is brood of different ages in adjacent cells. And this larva here seems to be forming a bit of a rubbery scale on the bottom of the cell. And perhaps this larva is a bit discolored or brown. So how can we confirm disease? So the most common way is to do bacterial culture. So we swab affected larva, we plate that larva on a special media, and we incubate that plate under anaerobic conditions for 3 days, and resulting in these shiny white colonies, which are consistent with Melissococcus plutonius. Under the microscope, these colonies are gram-positive or blue, and they form short chains of cocci. We can further characterize these bacteria using genetic sequencing, and EFB has sort of 3 groups of strains. And so these groups are shown here, and the colors represent the countries that these strains were isolated in. So on your left are the so-called atypical strains, and on your right are the more typical strains. So the atypical strains are thought to be the most virulent. They kill the larvae the most quickly in the laboratory. Whereas the typical strains are either non-virulent, so they don't kill larvae, or just somewhat virulent. And so you can see by the colors that most of the atypical strains are either brown or blue, indicating that they were isolated in either Japan or the UK. So this summer we obtained a disease larva that was taken from a colony in blueberry pollination and provided to us by Patricia. I'm getting a lot of feedback here. Patricia Wolf-Vega of the National Bee Diagnostic Center. And we cultured this larva for EFB, and interestingly, we isolated an atypical, highly virulent strain from this sample. And so this is the first report of an atypical EFB strain in Canada, and it was most similar to a strain that was previously isolated in the Netherlands. So this is very interesting to us, and I think it speaks to the fact that we need to do more surveillance in Canada to determine what strains we have. So again, we know that European foulbrood can arise in many situations, but if we use the situation of blueberry pollination as an example, what are some of the predisposing stressors which can result in an outbreak of this disease? So typically when we think about disease outbreaks, we think of 3 main predisposing factors. So host factors, pathogen factors, or environmental factors. And so if we think about blueberry pollination, one of the environmental factors could be pesticides. We know that fungicides are commonly used in blueberry production to control disease, and we know that pollinating bees will be exposed to these fungicides. And in the scientific literature, there is evidence that pesticide exposure can result in immunosuppression. We also know that there are different strains of EFB, and we demonstrated that highly virulent atypical strains are present in colonies pollinating blueberries. There are also host factors. So we know that blueberry pollination typically occurs in the early spring when colonies are often weak and may not have an adequate nursing bee population to take care of their larvae. As well, nutrition is another environmental factor which can predispose to disease. It's been published that blueberry pollen is low in protein, and we wonder if poor nutrition could be contributing to decreased resistance to disease. So if we want to study EFB further, we need to develop a laboratory model that will allow us to explore the influence of some of these predisposing factors. So our first objective was to establish a laboratory model of European foulbrood. And then once we have that model, we want to investigate whether pesticide exposure would predispose larvae to develop EFB in the lab. So to establish this model, we have the ability in our lab to grow honeybee larvae from a newly emerged larva into an adult in an incubator. And so what we did is infect larvae with Melissococcus plutonius, the strain that we obtained from BC, and then grew them in the lab. So on the top here we have a larva at day 10 of development, which is a control. And below we have a larva which has been infected with the BC strain of EFB. And you can see, even though it's the same age, it is much smaller, suggesting that it is unhealthy. And so we used histology to confirm that our laboratory infection was successful. So this is a microscopic section of a larva with the head and the midgut. And you can see that the control larva's gut is relatively empty. But our diseased larva has a mass of blue bacteria within its gut, which is consistent with Melissococcus plutonius, confirming that our laboratory infection was successful. So now that we have this laboratory model of the disease, we want to investigate the hypothesis, will larvae be more susceptible to EFB when exposed to pesticides? And specifically, we wanted to look at the insecticide thiamethoxam, which is a neonicotinoid insecticide, as well as several fungicides that are used in blueberry production, as well as the combination of thiamethoxam and a fungicide. And so what did we do? So we took newly hatched larvae from the field and grafted them in the laboratory and infected them with a pure culture of Melissococcus plutonius, the strain that we isolated from British Columbia. And then we raised them in the lab for a week and exposed them each day to either control or pesticide contaminated diet. And so the diet contained one or more of these pesticides. And we simply monitored survival every day. And so by the end of a week, we would have a plate of larvae like this. So the green box here shows the control group, which are nice and healthy. And the red box shows the group that's infected with the bacteria. And there are some small, unhealthy looking larvae here. And so using this experimental approach, we can generate graphs such as this. So the y-axis here is percent larval survival over 1 week. And the x-axis is the number of bacteria administered to those larvae on day 1 of the experiment. The colored bars represent the diet that these larvae were given. So the control uncontaminated diet, and then the red and blue bars represent 2 different doses of the insecticide thiamethoxam. So when we give the larva no bacteria, we see excellent survival. Between 80 and 100% of the control and pesticide exposed larvae survived. When we give the larva 50 bacteria, we see survival drops to 50 to 60%, but there's no significant difference between the control and the pesticide exposed larvae, suggesting that pesticide exposure did not predispose these larvae to die. And similarly, if we give more bacteria, 250 or 500 bacteria, survival drops 75 to 100% mortality, but there's no significant effect of pesticide treatment. So this data would suggest that thiamethoxam does not predispose larvae to develop EFB. So what about the combination of thiamethoxam and a fungicide? Could that predispose larvae to develop disease? So this is the same type of graph. The red bars here represent a fungicide, propiconazole, and the blue bar here is the combination of thiamethoxam with propiconazole. If we give the larva no bacteria, interestingly, the fungicide on its own significantly reduced larval survival relative to the control group. And this is a field-relevant dose of fungicide. So on its own, the fungicide might be slightly toxic to the larva. But if we give the larva bacteria, so 50 bacteria, there's no significant difference in survival between the control and pesticide-exposed larva. And the same thing holds true at higher doses of bacteria. But you may notice from this graph that the blue bars representing the combination of pesticides, they are shorter than the other 2 bars at each dose of bacteria. So it seems that there's a trend towards reduced survival of the larva exposed to the combination of pesticides and infected with bacteria. But it's not statistically significant in this analysis. But there is another way that we can analyze this data. So if we just take the data for these 3 bars here, representing larvae infected with 50 bacteria, and analyze them a different way. So here on the y-axis we have percent survival, and on the x-axis we have time, so day 0 to day 7. And all groups of larvae start off at 100% survival, and then survival slowly goes down as the experiment continues. Using this analysis, the blue line here, which represents the larvae exposed to the combination of thiamethoxam and a fungicide, had died significantly faster than the control larvae, which are represented by the black line. So this analysis would suggest that combined exposure of a larva to an insecticide and a fungicide predisposes it to die from European foulbrood. However, the caveat is that the concentration of thiamethoxam used in this experiment was 272 times environmental concentrations, so not realistic. This is common in research that we use unrealistic concentrations, which is sometimes frustrating to people listening to us. But it's important that we use a high dose so that we can understand what effect we are looking for when we gradually titrate and use lower doses. So what can we conclude from this data? Well, we successfully established a laboratory model of European foulbrood. And using this model, we have data to suggest that insecticide and fungicide exposure may increase mortality from European foulbrood, but more research is needed at more environmentally realistic doses. And luckily, we have acquired funding to continue this research. So we applied for a grant in collaboration with Patricia Wolfega and Marta Guarna, and received money from Project APSM and Costco to continue this research and to test additional pesticides and doses on larval survival from EFB in the laboratory. Also, as part of this research, we intend to collect and compare field isolates of Melissococcus plutonius to compare the virulence of strains in the laboratory. And so that's part of the reason I was handing out swabs at this meeting, to hopefully collect some field strains from Canada. As well, we know that pesticides are not the only thing which could potentially predispose to EFB, and we want to investigate the influence of nursing bee population and pollen nutrition on susceptibility to EFB. So as I mentioned, strain virulence is a pathogen factor which can potentially predispose to EFB. And we know that the virulence of different strains of EFB varies. So last summer, I did some preliminary collection of different strains of EFB from Western Canada. So I had several strains from British Columbia, one from Alberta, a few from Saskatchewan, and I had someone mail me one from Ohio. And all I did was grow these strains in the lab and then infect larvae with 100 bacteria of each strain. And so this graph here has percent larval survival on the y-axis, so that's survival over 7 days. And then we have our control group here, which received no bacteria and had excellent survival. And then the colored bars are representing the different strains, and each strain, we gave 100 bacteria of each strain to each group of larvae. And so you can see that the survival varies. Some strains here have almost 100% survival, are essentially avirulent or non-pathogenic, whereas other strains are killing 75% of the larva or more. So we want to investigate sort of the genetic features of these strains, which might contribute to their different virulence, and possibly correlate virulence in the laboratory to clinical virulence in the field. And so that's where I could definitely benefit from your help. If you have an outbreak of EFB, please contact your provincial apiculturist, and they can potentially submit samples to me. I am not a diagnostic lab. If you want diagnostic quality testing, you need to submit those to Patricia or the National Bee Diagnostic Center. But certainly my interest in the strains lies in understanding the different variants that we have in Canada and the different behaviors that we can see in the lab and the field. As well, we're also interested in moving from the laboratory model to a field model of EFB. So as I mentioned, there are host factors such as weak colony strength, as well as environmental factors such as pollen nutrition, which could also act as stressors and predispose colonies to EFB. So my colleague in the lab, Dr. Ivana Kozy, will be working this summer to develop a field model of EFB to investigate those two potential stressors. So in her work, she will be establishing experimental colonies which have a laying queen in the bottom chamber and then two experimental compartments in the top brood chamber. And these experimental compartments will contain pollen sourced from either blueberries or prairie pollen. And this pollen will either be provided as a patty or as a frame of bee bread. As well, within these experimental compartments, she will be varying the number of nursing bees. So some compartments will have a large number of nursing bees, and other compartments will have a small number of nursing bees. And she will be infecting the larvae in these compartments with EFB strains, and then comparing survival of these larvae in the different compartments with the different pollen and nursing bee treatments. And I'd like to acknowledge Frank Shimonuk. Is he in the audience? He is. Okay, well, Frank, I owe you a beer. I almost bought a beer for another Frank at this conference because I thought he was you. But Frank has kindly offered to provide us with some frames of blueberry pollen for this field experiment. And so we're very grateful for his contribution. So the overall goal of our research is really to change headlines like this so that blueberry fields do not pose a health risk to bee. But you can insert other conditions there too. Really, we just want to minimize the occurrence of this disease in the field. And so by better understanding the predisposing factors to outbreaks, we hope to mitigate these outbreaks. So I'd like to acknowledge my lab, as well as Patricia Wolfega, Marta Guarna, and Frank, as well as the many funding sources that we have for this research. And this is my contact information, and I'd be happy to entertain any questions. Thank you.